Hello friends, welcome to my channel. Today we are going to discuss about the Anther culture. In this topic, we are going to discuss about introduction of Anther culture, objectives of Anther culture, what are the requirements to carry out the practical of Anther culture, procedure and applications of Anther culture. So before moving towards the Anther culture, we all know about the tissue culture. So tissue culture, it is the branch in which you can take any plant in any season. You can get more quantity, more yield, you can induce the mutant plants, you can produce the genetically modified plants by using that tissue culture. But this tissue culture is based on one of the process that we called it as an totipotency. So what is the totipotency? So it is the ability of a plant cell to divide and redivide to produce the whole plant. So for this process, the plant part which we select that we called as an explant. So just see here that what is the anther culture. So anther culture means whenever we are going to produce the plant from the anther, or you can say that whenever we are going to use anthers as an explant, so that type of culturing we call it as an anther culture. So anther culturing, it is basically going to carried out for maintaining the haploid cultures. So haploids refer to those plants which can possess the gametophytic number of chromosomes in their sporophyte. So these haploid plants, they are going to categorized under two categories, that is monoploids and polyploids. So monoploids means the plants which possess half the number of chromosome from their diploid species. So that we called it as an monoploids. Then what is polyploids? So polyploids are the species which possess half the number of chromosome from a polyploid species. So just consider here one of the most important aim to carry out the anther culture is that to maintain the pure lines, or you can say that to maintain the haploid cultures. Then we see what are the requirements to carry out the anther culture. So here we require the anthers from any flower. So here I will take the hibiscus. You can take any flower or anthers of any flower. So for the growth of that plant we require the media. So here we are going to use the MS media, which is most popular in the tissue culture. We require different types of growth regulators, growth supplements. One of the important step in the tissue culture is that sterilization. Okay. So in any experiment of tissue culture, there is a proper sterilization of glasswares. There is a proper sterilization of media that you have used. There is a proper sterilization of the surface area on which you are going to work. There is a proper sterilization of explant. So for that sterilization process, here we require 70% ethanol, then 0.2% mercury chloride. According to need, we can use twin 20 or twin 80, then mesoinositol. So these are the various chemicals which we are going to use for the sterilization purpose. So here before carrying out this experiment, just see here or just remind that your all glasswares what you need to perform that experiments, all of them they should be sterilized one by using autoclave or by using hot air oven. You can sterilize them. Then whatever the forceps scissors, then if you need the distilled water for that one, so it should be properly sterilized one. Your working area you have to clean by using 70% ethanol. Your hands you have to sterilize. Then the next thing is that before carrying out this experiment or before the inoculation process, you have to on the UV of laminar airflow for the proper sterilization. Okay, so in this way you have to carry out the sterilization. Okay, then next moving towards next step, that is the procedure. So here how we are going to perform that anther culturing. So he see here that firstly you have to collect the buds of flower. Here I will take the hibiscus. You can take any flower. You have to collect the flower buds. See here, as you are going to perform the anther culture, so you don't take the anthers and sterilize them. Here what you have to do, you have to collect the flowering buds and you have to sterilize them. And after the proper sterilization, then you have to remove the anthers. So just remember this one. So even though we are going to carry out the anther culture, we have to dip or we have to give all the treatments of sterilization to the flowering buds. Okay. Otherwise our anthers they get injured one. Okay. So first here see that the flowers firstly you have to rinse with the distilled water. After that you have to immerse that flowering buds into 70% ethanol for 60 seconds. After that the next step is you have to remove that flowering buds from the 70% ethanol and dip them into the 2% sodium chloride solution just for 1 minute. Okay. uh, if you are going to increase the time, then they will damage the anthers. That's why uh, here the time is very important. You have to just keep that flowering buds only for 1 minute. And after the treatment of HgCl2, just you have to remove that floral buds and you have to thoroughly that wash that floral buds by using the distilled water several times to remove all the traces of mercury chloride. Okay. Then after you have to transfer that buds into the sterile petri dish. Okay. Then after you have to split the bud, okay, by using the blade, and you have to remove the anthers, okay, without damaging them. You have to remove the filament, just you have to collect the anthers. Okay. Then already you have prepared the MS media. You can make the slants of them, or you can inoculate them into the culture bottles, which are basically used in the process of tissue culture. So you have to inoculate the anthers horizontally on the MS medium, and see that all this process you have to carry it out under the laminar airflow. Okay. Then in our MS media we are going to add the growth regulators like auxin, cytokinin. Then you can add the mesoinositol. Okay. After that you have whatever the type of if you are going to use the slants or petri plates or whatever the culture bottles, so depending upon that you have to seal them properly. Okay. And firstly you have to incubate them in dark at 28 degrees Celsius. Okay. And you have to check them for germination of anther. And whenever there is a germination can takes place, you can transfer that anthers into the or the subculturing can takes place, in which you are going to use the same media, but you can increase or decrease the amount of growth regulators depending upon your requirement. So just see here, this is the diagrammatic representation. Here you have to remove the floral buds. See here, firstly you have to wash them with distilled water. Then you can wash them by using 70% ethanol. Then you have to dip them into the 1% HgCl2. Okay. After that you have to thoroughly wash that floral bud several times with distilled water to remove all the traces of HgCl2. After that you have to take that floral bud into the sterile plate, petri plate. You have to use the sterile scalpel to break the floral bud, and you have to just see here, you have to remove the anthers. After removing the anthers you have to also remove the filaments. So see here, these are our sterilized anthers. Okay. and that sterilized anthers you can inoculate them into the petri plate. Then you can observe their germination, and after their germination you can transfer them into the culture bottles, slants, or petri plates, whatever you are going to use for the process of anther culture. So just see here that after 2 to 4 weeks you are able to see the plantlet. Okay, if the sterilization conditions are properly maintained one and the composition of media is proper one. Okay, then next see that whenever we are going to carry out that experiment, actually this is our floral bud. See here, after proper sterilization you can cut them. These are the anthers. That anthers firstly you visualize for its germination. After germination you can subculture them, and after subculturing you are able to get the plantlet. That plantlet you can harden and keep them under the soil, compost, uh, and coco peat, 1:1:1 composition under the greenhouse. Okay, so in this way the process of anther culture is going to be carried out. So see that what is the importance of anther culture, why we have to carry out the anther culture. So the main aim is that to obtain the haploids, then to study the genetic recombination in the plants. By using anther culture we can easily check them. Then the study of factors which can control the pollen embryogenesis, we can study it by using the anther culture. Then to produce the homozygous plants, basically this anther culture it is used for the alkaloid production, for the development of hybrids. We can study the different types of mutations. Okay, by using anther culture. Then we can produce the disease and insect resistant plant by using the anther culture. Okay, we can get the higher yield. Okay, so these are the several importance of anther culture. So in this way the anther culture is going to be carried out. So if you like this video, please like, share and subscribe my channel. Thank you.